Hi class, Dr. Jim here. So in this video, this is the second part of the two. When we talked about transcription in the first video, this is translation. And so if you haven't watched transcription yet, do not watch this video because it will make you more confused if you haven't seen the first one. So if you haven't seen transcription, watch that one first. For those that have watched the first video, welcome to the second video in this process. And what this will allow us to do is now go from the first step, which was transcription, to the next step, which is now going from mRNA to protein, and that's called translation. And in this video, we're going to explain a little bit more about how this process actually works. We're going to take you through the steps, what everything looks like, and where this process takes place. Okay? So let's begin our lecture. So the objectives for this is first, what is the process of translation? And I've got a great explanation for this. Told you before how we talked about writing notes and things like that in the first video. In this video, I've got another great explanation, and hopefully it really helps you get the point of what's going on. The second thing we're going to do in this process is actually look at what is the process. Where does it take place? So translation takes place outside the nucleus in eukaryotes. In prokaryotes, since there is no nucleus, it can all happen in the cytoplasm. But in eukaryotes, we did transcription in the nucleus. Now translation, the second step, going from mRNA to proteins, takes place in the cytoplasm. And in the cytoplasm, we have these things called ribosomes, either associated solely in the cytoplasm as free-floating, or those associated with the rough ER. So it's one of those two areas, and that's what we're going to look at. The other thing is how does it all work? And I'm going to take you step by step how translation takes place and what actually happens in those situations, okay? And so we'll go through those things and we'll see what happens. All right. So translation. What is translation? Translation is the sequence of codons in the mRNA at the ribosome that directs the sequence of amino acids. And so basically what it's saying is that now you're going to go from one language to another. All right. So let's use my note example that I used in the last video. Okay. We talk about our notes. What I mean by that is, let's say you remember you missed class and now you have to borrow someone's notes and you made an exact copy with slight subtle differences. Well, that's what transcription is, making a copy. And this time, now what you have to do, let's say you have a roommate that doesn't speak any English and they want the same notes you just copied. Let's say they missed class 2. But now you have to translate it from the English that you have into a totally different language. Let's say like English to Spanish, or English to French, or English to, let's say, some alien language like Klingon or something like that. Whatever your language that you want to translate into, that's your business, all right? But anyways, you have to translate it, and that's what we're doing here. We're going to go from an mRNA or nucleic acid language into a protein language, and so we have to translate it, and that's the second step. Basically what happens is that we take our mRNA and our A's, G's, C's, and U's and convert it into a protein language, and I'll show you how that happens in just a minute. What you see is the nucleic acid is translated into a protein sequence, and that's what I mean here. You can kind of start to see what we're dealing with, and no longer are we worried so much about the individual letters, but what we're going to be worried about is groups of letters, the groups of 3, and we're going to talk about that in just a second. Okay? So if we look at the genetic code, the genetic code is consisted of codons. And so let's take the mRNA language. We talked about mRNA and it being a language, right? mRNA is a language, okay? Has these letters, A, G, C, and U. Now what you want to do is start putting those letters into words. And in your vocabulary, all you can do is make combinations of 3-letter words. Okay, so only words that are in your language, in your genetic language, is going to be 3-letter words, and that's what a codon is. So no longer are we worried about just the A's, not just the G's, C's, and U's, but now we're worried about AUG, or GGC, or CGA, or UGC. Okay, those are the words now that we're concerned with. And so if you look at this, we have 20 amino acids. So in our bodies, between bacteria and humans, we have 20 amino acids that make all our proteins. Okay? And in order for that, it's specified by these codons, these words. And so we have the 4 letters, okay, the mRNA letters, A, U, G, and C. And now what we're worried about is the words, making all these different 3-letter words. If I gave you those 4 letters, and I told you to make as many 3-letter words as you could, you would come up with a total. If you spent the whole time, you'd probably get frustrated by the end. But if you spent your whole time making those words, you'd come up with 64 words. Okay, so that's the nice thing, is that this language is actually pretty small. There's only 64 words in the whole or in the whole vocabulary of genetics, and so that's nice. So we look at that and we talk about the triplets, or the 64 words that are involved in the genetic code. Now, why is this important? Well, each one of those words codes for a specific amino acid, okay? And that amino acid could be any of these different codes based on whatever we see here. Now, this is called a codon usage chart. And so what that means is that you can look at any combination or any of those words and find out what amino acid it means. This is basically the dictionary to that alphabet. Okay, or that word. So let's say you have CCC. Let's say we're going to look for CCC. How would you use this codon usage chart? Well, first I'd come here to what is called the first base, and I would look for the C. So I'm looking for the word CCC. So I found my first C. Okay, then I go to the second base, which is on top, and I look for the second C. And so I know I go to this column. So I start here and go here. And then I look for the last C, which is here. And so I look for my word CCC, and I see, CCC means proline. And so my codon, that word CCC, means proline. And I can do this for all 64 words. I can come up with a different amino acid for each one of these words. Okay? I'm not going to ask you to memorize these things. I don't know all the codons and what are used for. This is why we have a chart. Okay? The one that I think you should look at and know is AUG. AUG is always the start, or the start of where translation takes place. And so AUG is always an important spot. The next time you come to lab, I want you to look at my button that I have on my name tag, and I want you to get the idea of what that actually means. Okay? So if you read that and you see what that button, I want you to kind of put it together and see what that means. And if you can figure it out, you get the A for the day. Okay? So remember, AUG is the start. We also have 3 stop codons where things stop, and it tells the ribosome you stop here. Okay, and then all these other ones have different amino acids associated with it. And so again, this is very useful when you're trying to figure out what does the word mean. Okay, here's your dictionary for your codons. All right. So what about the genetic codons that are important? Well, first of all, it's universal. So that CCC encoding for proline is the same in bacteria that it is in for us. Okay, so CCC means proline in bacteria, and CCC means proline and in us, and so it doesn't change, and that's a nice thing. That's universal. The other thing is that they're redundant. Now you know there's 64 words, but I told you there's only 20 amino acids. Okay, so that means there's some codons that get used for more than one amino acid. Okay, so the codon, or what I should say is that some amino acids have 2 or more codons associated with that. Okay, and that's important. It's redundant. So if you have a slight change in your letters, let's say you go from CCC and go to CCG, it means proline, the same thing. Okay. Other thing is that it's unambiguous, which means that, again, CCC only means proline. It will never change to glycine. It will never change to alanine or any other amino acids. CCC will always be the same. And so you can always be sure when you look up CCC, whether you're talking about bacteria or humans, is always going to mean proline as the amino acid. All right? The other thing is we always have a stop and start signal. And so we have to know where things start and where things stop. You always, in a good book, have the start, the middle, and the end. And the same thing works with mRNA. You always have the start, the middle, and then the end. Okay? And so that's also important. All right. So if you remember my transcription video the last time, I talked about 3 different types of amino acids. I have 5 here, but I mean 3 types here. You have 3 types of RNAs. You have the mRNA, which is the messenger, and we talked really about that in the transcription. And then there were 2 others. There was the tRNA and the mRNA, or the rRNA, and I told you to worry about that for this video. Well, here we go. tRNA means transfer RNA, and that is going to transfer the amino acid over to the ribosome. So you're transferring it over to the ribosome. The other one is the rRNA, and we're going to talk about that in just a minute. That is the ribosomal RNA, and it's involved in the ribosome, and we'll look at that in just a second. The interesting thing about the transfer RNA is that it has two parts that are really important. The first part is where the amino acid binds on the top, and it always binds the same amino acid each time. And then on the bottom, it has the anticodon. And the anticodon is like the evil stepbrother. If you remember watching 80s TV shows or 90s or maybe soap operas, where you'd always have the guy that is the same dude, but he has the mustache. Well, that's basically the same thing. You have the codon and the anticodon. The anticodon is the opposite of the codon. So if we have a codon of CCC, our anticodon is going to be GGG. If you have a codon of AUG, you're going to have the anticodon of UAC. Okay? I'll give you an example. You tell me what the anticodon is. If I have one that is GCU, what is the anticodon of GCU? That's right, CGA. Okay? So it's always the opposite. All right? So remember that. Always the opposite of whatever my codon is. And again, this is something that you just need to practice. If you get it right away, you're going to work on this in lab, but we'd like you to know the opposite. All right? All tRNA molecules have the amino acid and have an anticodon. And here's the structure of the tRNA. Here's the amino acid binds, and here's the anticodon. Okay? Anticodon always is the opposite of the codon. And you can see here, CUU binds to GAA. So let's do this one. This is the anticodon. CCU, what is the anticodon? That's right, GGA. Okay, so if you got those, you're doing really well. Okay, and then each time this tRNA molecule binds the same amino acid. So this specific anticodon will always bind the same one, leucine. Okay, and that's how we'll look at it. Now the other important part of this process is the ribosome. So this is the location. You remember me talking a little bit about ribosomes a couple minutes ago? Well, ribosomes are involved in making the protein. This is where translation takes place, and it can be either in the cytoplasm or in the rough ER. Okay? And associated with those ribosomes is another brand of RNA, and this is called rRNA, and we call it rRNA because it's ribosomal RNA. And this is the last time we talk about it, and it's basically formed and made into these different subunits of the ribosome, and it helps move things along and build the structure. The other important thing about ribosomes is that there's actually 3 sites inside the ribosome that are important for binding the tRNAs, and that's called the E, the P, and the A. The E stands for exit, the P stands for peptide, and the A stands for the amino site. Okay, and we're going to look at all these things in just a minute, so if you don't get it yet, we're going to see some visuals, and I'll show you. Okay. So here are my 2 subunits. I have my small subunit and my large subunit. Okay, the large subunit, it kind of looks like a big walnut kind of like structure. The mRNA fits into this groove here. So if I get my pointer back, here we go. It fits into this groove right here. So this is where the mRNA. So think of an old film or ticker tape or something, and it fits into there and it kind of clamps down on it. You can see here, here's the mRNA and how it comes in. Now, inside the ribosome, you have these 3 sites, the E, the P, and the A, and that's where the tRNAs bind. And you can see here, this fills into the P site. The P site is in the middle. Then the next RNA comes in, tRNA comes in. You get this peptide bond that forms between the amino acids. This shifts over, goes in the exit site, the tRNA comes out, and now the next tRNA comes in. And this is how the process of translation works. It builds like this. And you can see you can actually have multiple ribosomes on one mRNA. And so this can be going all the time. And this is how you make lots and lots of protein very quickly. All right? So translation. We're going to talk about initiation, how things begin. And so initiation is the process of starting. And so what is the start codon we talked about? That's right, AUG. So what happens is the small subunit looks for the AUG on the mRNA and binds with it. Then the anticodon comes into that. So AUG, what would be the anticodon? That's right, UAC. That binds onto the tRNA, or the tRNA binds onto that codon with the anticodon, and then the large subunit kind of clamps down on top of everything else, and that's what we're talking about here. So here you go. Here's the visual for all you visual people out there. Here's the small subunit, okay? Looks for that AUG. Remember, AUG is always the start. Here comes the tRNA with the anticodon. See how it's the opposite? A's always go with U's, A with U, G with C, and so it's the opposite. Binds on, and then the small subunit binds on top of, or the large subunit binds on top of that. And once that starts, the process can begin, and that's called initiation. Okay, initiation comes together and starts the process. The second step is called elongation, and all that means is that you're going to elongate your protein or elongate your polypeptide by adding more amino acids. And so really what happens is that the tRNA, the next tRNA comes in, binds on, everything shifts over one, you lose the last tRNA, allows for the next tRNA to come in, and it's a process going boom, 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 and you have lots of booms, okay? And you keep elongating the polypeptide. And this is what happens. And so visually you can see here, I have my tRNA, let me get my pointer back, in my ribosome here. It binds with the anticodon, okay? So here's my codon, GUA, the anticodon CAU. The next one comes in, here's GAC, so my anticodon is CUG, that binds in. You attach the amino acids together with the with my peptide bond that forms the bond between the two amino acids. You can see it right here. Soon as that bond is made, everything gets shifted over one. This tRNA ends up in the E site, it leaves, and now you have the open spot for the next tRNA to come in, which is ACC. The anticodon is UGG, comes in, and you now elongated the polypeptide, and that's called elongation. All right, so let's look at the last step, which is called termination. This is when you come to a stop and how the ribosome knows where to end. And so basically it comes to the next site. It encodes for one of those three stop sites. The mRNA has the next of those three words that mean stop. Comes to that spot. There's no corresponding tRNA for those codons. And so those three stop codons have no corresponding anticodon that can bind to it. And when that happens, it says, okay, there's nothing to fill in the spot. That must mean we come to an end. The ribosome comes apart, the protein is released, and the process can start all over again. And let me show you visually. So we come to the end. Here's the stop codon, UGA. UGA, there is no anticodon for that. There's no anticodon, so this release factor fills that spot. And since that is there and no tRNA, you can't build a peptide bond to the next amino acid. Everything falls apart. The tRNA leaves, the mRNA comes off, and the ribosome split apart. And now the protein can go off and fold to whatever that protein looks like. Okay? And so that process is called termination. And so you can put all three of these processes, all of these are called translation. You start with initiation, which is the start, elongation, and then termination, which finishes the process. And this next slide just kind of shows you the whole process from transcription all the way through the end of translation. So if you remember transcription, we did that in the DNA. We made the copy, or the nucleus. You made the copy, you process, and you made the mRNA. The mRNA comes out. The ribosome looks for that start site, the AUG. Once it finds it, it binds on. Here's the initiation, binds on. The tRNA comes on, binds with it, and then you get the elongation. You go through that process and you elongate your polypeptide by constantly adding new amino acids and peptide bonds. Once you come to the end that finds the stop codon, that stop codon says, okay, time to stop. Everything gets released. The mRNA comes off. The protein gets released inside the ER if that's where it's getting made. The small ribosome and the large ribosome subunits come apart, and the process can begin again. And that's the step-by-step process of translation. Okay? Highly recommend watching this video. It's a really good video. It will take you through the whole process through initiation, elongation, and termination. And again, it will show you graphically in a video, and it'll probably even make more sense than me explaining to you. So make sure you watch this video. All right, and so we come to the end. Translation. Translation is the process of translating the nucleotide sequence into protein words. Okay, so we translated each one of those letters, A, G, C, and U, into now words that mean something. Remember, each word, each three-letter word means a different amino acid. So CCC meant proline. AUG means methionine, and that never changes. It's universal throughout. So never changes from bacteria onto us. Okay? We also make peptide bonds between the amino acids and grow our proteins and make them bigger and bigger and bigger. And then finally we reach the stop codon where the whole process stops and you start again. All right, and with that, we come to the end of this lecture. If you have any questions, please send me an email. Please talk to me in class. We'll find out what your issues are or whatever your questions are. Please let me know. And with that, I'll be happy to take any questions that you might have, like I said. And please really work on this in the lab. I think you'll actually get something out of it this week and get something that goes. And for that, I'll see you next time.